Today, I'm going to talk about the multiphase roles of neutrophils in cancer and very briefly introduce our group's work on herbal medicines regulating neutrophils within the tumor microenvironment. 1.3 million people are estimated to be diagnosed with cancer every year. Robert Weinberg, the cancer researcher, says, if we lived long enough, sooner or later, we will all get cancer. As science advances, we get to live longer and healthier, but still, cancer is a problem that has not been solved. Surgery was the only available option before 1955 when the Curies discovered X-rays. And since then, the anti-cancer therapies have gone through three more major steps. Chemotherapy, targeted therapy, and now we are currently living in the era of immunotherapy. In the year 2000, Douglas Hanahan and Robert Weinberg, again, published a paper in Cell, The Hallmarks of Cancer. In this review article, which is cited over 40,000 times, the authors suggest 6 hallmarks, 6 characteristics of cancer which make cancer like cancer. Cancer cells avoid apoptosis and anti-growth signals and try to grow through self-growing signals, and they produce angiogenesis and et cetera, et cetera. These people later update their view on cancer in 2011. This review paper, also published in Cell, added 4 more characteristics of cancer. This time, they say that the immune system is quite important in cancer. So when people started to focus on the immune system, they realized the importance of the tumor microenvironment. The tumor tissue does not only include cancer cells, but have so many other components forming an environment. Look at these cells. The tumor microenvironment contains cancer cells, fibroblasts, immune cells, extracellular matrix, blood vessels, and so much more. These components play their own roles, and since the nature of cancer cells is quite smart, the tumor microenvironment turns into one that makes cancer grow and migrate better. Among these components, today I'd like to talk about neutrophils. I personally have particular interest in neutrophils and their role within the tumor microenvironment. Our lab's work involves several diseases such as allergy, sepsis, obesity, and cancer here, but there's one common rule in our lab despite this broad spectrum of disease models. We focus on the role of neutrophils in the pathogenesis of these diseases and try to use them as a therapeutic target. So, what are neutrophils? Neutrophils are the most abundant leukocytes in the blood and responds first of all immune cells to alien pathogens. In response to infection or inflammation, circulating neutrophils display surface molecules that facilitate their interaction with the activated endothelium. So through this way, the neutrophils migrate to the site of infection and inflammation and kill pathogens or dead cells by methods like phagocytosis or mitosis. This is their most important function, the first defender in the innate immune system. They also communicate and interact with other immune cells like B cells, T cells, dendritic cells, natural killer cells, and therefore contribute to adaptive immunity as well. Neutrophils have long been used as a marker to predict the prognosis of cancer patients. The neutrophil to lymphocyte ratio is an efficient predictive marker of the prognosis of cancer. Other than this, the neutrophil to lymphocyte ratio, neutrophils did not gain much interest in the field of cancer. Early studies on neutrophils in cancer have shown that neutrophils are related to poor prognosis in cancer patients. The tumor growth induces infiltration of neutrophils, which were called tumor-associated neutrophils, in short, TANs. These TANs were known to suppress anti-tumor immunity and therefore promote the tumor growth. One specific mechanism of this was involving the well-known immune checkpoints, PD-L1 and PD-1. Neutrophils, especially the ones infiltrated into tumor sites, express high PD-L1. This PD-L1 then binds with PD-1 of T cells, preventing T cells from recognizing and killing cancer cells. That's one example of how neutrophils allow tumor cells to avoid the immune system. But recently, the diverse role of neutrophils in the tumor microenvironment have been discovered, and the importance of neutrophils in cancer is gaining much interest. There are currently recent advances in neutrophil research discovered there are two types of neutrophils, the anti-cancer N1 neutrophils and the pro-tumoral N2 neutrophils. N2 neutrophils are the dominant phenotypes shown in cancer patients, and these N2 neutrophils make cancer more aggressive, more metastatic. So the previous introduced high PD-L1 expressing neutrophils was found out to be one of the action of these N2 neutrophils. But N1 neutrophils, on the other hand, have anti-cancer properties. Especially this subtype called N1A type neutrophils have exceptionally high cancer-killing abilities. So in short, later discovered knowledge says that depending on the neutrophil phenotype, it can be pro-tumoral or anti-tumoral. This suggests that the role of neutrophils is quite complicated and still not fully defined. It also suggests that the polarization of neutrophils towards the N1 phenotype is a possible strategy for anti-cancer therapy. So as you can see here, the recent advances in neutrophil research are now establishing the concept of N1 and N2 neutrophils in the tumor microenvironment. Also, there are currently ongoing clinical trials targeting the neutrophils for cancer treatment. This also supports the significant role of neutrophils in tumor progression. So our lab's current work greatly involves neutrophil extracellular traps, in short, NETs. Originally as a defense mechanism to kill pathogens, NETs can lead to other consequences. For instance, excessive NET formation due to a haywire of immune response can damage nearby tissues. Or since NETs capture pathogens or dead cells, they can also capture other cells alive. And when they catch platelets in the circulation system and aggravate, this can cause a clog and block the blood flow, which means ischemia and stroke. But NETs can also capture cancer cells and facilitate metastasis by carrying these cancer cells to a remote area. And NETs also induce the shift of macrophages or suppress T cell activity in the tumor microenvironment, leading to a suppression of anti-cancer immunity. So one of our group's recent work involves a chemical drug named disulfiram. Disulfiram is a drug to treat alcoholism. It works as an ALDH inhibitor and makes the body not being able to get rid of acetaldehyde. Therefore, the alcohol consumer suffers from nausea, vomiting, dizziness, and so on. Disulfiram is the only FDA-approved drug that works as a netosis inhibitor. And this is why we selected this drug as our first candidate to use in our anti-cancer work involving neutrophils. Recently, a team in the Cold Spring Harbor lab, in collaboration with Mount Sinai and Cornell Stony Brook, showed that disulfiram can inhibit netosis and treat COVID-19. Although there are quite a lot of papers on the anti-cancer effect of disulfiram, there is not a single one that regards neutrophils and NETs. So we designed a study to evaluate its anti-cancer effects in non-small cellular lung carcinoma cells, considering the effect of netosis inhibition. So in line with previous studies, disulfiram, of course, suppressed tumor growth. You can see that disulfiram treatment group showed less volume or less weight in tumor size. But especially when focusing on neutrophils, we also saw a significant suppression in pro-tumoral N2 neutrophils along with decreased NET formation. Also, N1 neutrophils, the anti-tumor types, are notably increased. Further studies are currently ongoing, and we are trying to reveal a new anti-cancer pathway by using disulfiram. This may be a repeated result of the anti-cancer effect of disulfiram. However, this is the only study on its net-related anti-cancer effect. Also, these results allowed us to further keep on working with the importance of net formation in this allograft cancer model. So coming back to Korean medicine, there are several herb-derived compounds or herbal decoctions that can control and regulate and suppress net formation. Here, these are just a few examples on net-suppressing herbal medicines. SHO03 is a newly developed herbal formula combining these three herbs. Previous work done by our colleagues reported that the trichosanthes, the Chinese cucumber, exerts anti-cancer effects through the apoptotic alteration. And also the combination of astragalus and angelica shows immune enhancement. Thus, to provide anti-cancer effects while supporting the immune system of the patient, these three herbs were combined and studies were done to verify its efficacy. We analyzed through network pharmacology and discovered that there is a possible association between SHO03 and NETosis-related pathways. 12 genes playing critical roles within the formation of nets can be regulated by SHO03. Also, using freshly isolated neutrophils, we see a significant suppression by this herbal decoction. Here you can see the decreased citrullinated histone H3, which is an important marker in NETosis. And we verified this by staining the neutrophils and also using Western blot analysis. Also, myeloperoxidase, elastase, and P84 were decreased as well. Reactive oxygen species is a component of NET2, so we conducted a real-time analysis of forced ROS measurement, and SHO03 suppressed ROS release. Elastase here is also a NET component, and we see decreased elastase in a PMA-induced NETosis test. So moving on, we conducted an animal study because we wanted to see the role of neutrophils and their interaction with other immune cells in the tumor microenvironment. So we decided to use a mouse cell line instead of a human cell line in immunocompromised mice. LLC1 is short for the Lewis lung carcinoma, which is a cell line established from the cancerous lung of a C57BL6 mouse. After subcutaneous injection with these cells, we fed the mice with SHO03 via daily oral gavage and applied cisplatin by weekly IP injection. The mice were sacrificed at week 5 when the diameter of the tumor in the vehicle group reached around 1.5 millimeters. Cisplatin, the well-known chemotherapy, of course, decreased the size and weight of the tumor. As with previous reports shown before, SHO03 also effectively suppresses the tumor growth. Of note, the combination of SHO03 and cisplatin displayed a very promising synergism in tumor growth suppression. Such effect was due to altered apoptosis. This work is done by colleagues in Harvard Medical School, Dr. Hyo-In Kim. CyTOF is a relatively new analysis tool mostly used for immune cell analysis. We could see a suppression of PD-L1 expression in all 3 treatment groups, but the highest suppression was seen in the combination group of SHO03 and cisplatin. Also, CD66B, an adhesion molecule of neutrophils and also used as an N1 marker, was slightly decreased by cisplatin compared to vehicle group, but this was altered in the SHO3 group. Combination showed an expression in between. On the right, we used the N2 polarized neutrophil model. Treatment with cisplatin could not regulate the N2 markers, but SHO3 treatment decreased the expression of N2 markers, and also below, upregulated the N1 markers. Note that the N1 or N2 neutrophils do not lack a certain marker when polarized, but the phenotype is determined by the expression levels of these markers, whether they are high or low. So in the animal model, after sacrificing the mice, we stained the cancer tissue with Cytox green, a DNA stain, and also a citrulated histone H3. DNA and histone H3 both compose the neutrophil extracellular trap, along with some other enzymes like elastase and MPO. Netosis is also an N2 neutrophil-mediated process which can capture circulating tumor cells, carry them around, and thus promote distant metastasis. Netosis also activates tumor cell metabolism, favoring tumor growth. You can definitely see that SHO3 decreased net formation in the tumor. Cisplatin inhibits tumor growth, but did not do much on the net formation here. We also used flow cytometry with single cells isolated from the tumor tissue and checked the expression of netosis. Further flow cytometry was performed to analyze net formation in the circulation system as well. Net markers in the blood neutrophils becomes higher in tumor-carrying mice, and SHO3 showed a promising regulation in the net marker. Below is the ratio of CD8 positive cells in the total cells from the spleen. We could see that cisplatin, SHO3, and the combination all increase the killer T cells in the spleen, but still the activity of tumor killing of these CD8 positive killer T cells should be further determined. The hallmarks of cancer is constantly being updated. The most recent work by Dr. Hanan added further hallmarks of cancer in 2022. The human body cannot be understood properly without considering the huge community of microbial components. Microbiomes are everywhere in our body, in the skin, in the mouth, in the lung, in the gut, and also in the tumor microenvironment. The community of microorganisms living in the gut, the so-called gut microbiome, play an important role in human health. The host-microbe interaction develop the innate and adaptive immune system. Neutrophils, like any other immune cells, interact with gut microorganisms, and the crosstalk between them affect each other. This makes the gut microbiome important in cancer as well. Considering the involvement of gut microbiome in immune cell regulation, the immune checkpoint is also linked very closely. Gut microbiota is one of the important factors which decides the response to immunotherapy. So next, we analyzed the composition of gut microbes to see whether cancer can make a change there, which is already shown previously, and also what cisplatin or SHO3 or the combination of two does to the gut microbiome. Allograft of LLC1 tumor cells leads to disrupted gut microbiome balance. Cisplatin injection, yet it suppresses tumor growth, cannot change the microbiota imbalance, but SHO3 and the combination here restored gut microbiota diversity and balance. This, although requires further investigation, would be probably related to the anti-cancer effect of SHO3. Another important point I'd like to emphasize is the limitation of chemotherapy. There are certain side effects that cisplatin treatment drives, such as pathological immune cell decrease or body weight loss. Here we show that SHO3 does not only show such side effects, but actually helps that to recover when combined with cisplatin. The decrease of immune cells is a symptom of blood deficiency in Korean medicine, and Angelica, one of the components of SHO3, is a powerful blood tonifying herb in Korean medicine. Also, the weight and muscle loss can be considered as a result of pathological qi deficiency, and astragalus is a qi supplier. This study today is only a brief hint of the use of Korean medicine for cancer patients. However, given that there are several immune-enhancing herbal agents frequently used in Korean medicine, I dare to say that the potential of herbal formulas in Korean medicine will indeed be a great advantage in cancer therapy and suppressing tumor growth and improving sensitivity to anti-cancer agents, and also by alleviating side effects. Herbal medicines used in Korean medicine have many strengths, especially when we consider the brand new hallmarks of cancer. Natural products are fascinating agents which can control cellular phenotypes and plasticity. When considering the senescence of cells, quercetin, a famous flavonoid rich in a number of medicinal herbs, is one of the world's most promising xenotherapeutics. Epigenetic regulation is affected from the environment, including diet. Herbal medicine, largely intersecting with food, has great potential because of their epigenetic effects. And seen today, herbs can be a rich source of pre or probiotics and affect the human microbial community to treat diseases. Well, there might not be much for you to take home from my presentation, but if you may, please remember the interesting facts about neutrophils. Neutrophils, the previously unimportant immune cells in the tumor microenvironment, is gaining more and more interest in this field. A number of researchers, including myself, are focusing on their roles and how they impact cancer growth and metastasis. The complicated network lying in between neutrophils and the tumor microenvironment is very complex, but this makes neutrophils a highly attractive therapeutic target. There is so much potential of herbal medicine in this field, considering their action on immune cells, and I believe this is an amazingly exciting research. The work presented today was able by my lab members, Dr. Kim, Dr. Han, Ms. Bu, Mr. Sim, Ms. Kim, and our recently joined undergraduate researchers helped us a lot too. All these collaborators have contributed to my research, and I would like to thank them as well. Thank you.